So what we're going to do is we're going to work through these different processes, and we'll keep coming back to this summary to remind ourselves of how far we are on the journey of how a plant reproduces. We've got gamete formation, making the pollen, making the eggs, pollination, which is how the male gamete gets the female one, then fertilization when they fuse together, then you make the seed and the fruit, then you disperse that seed, and hopefully then you get the germination and the offspring being fully produced. Once a seed has been dispersed, that seed is actually in a dormant state. It won't start growing any further into a proper plant unless all the conditions are right. We call this process germination. So a seed only contains about 10% water, but if you really increase the water levels and also give some oxygen and also make the temperature nice and warm, the seed will germinate. So you need water, temperature and oxygen for germination to occur. The seed contains the little embryo, the little root shoot, and one or two what we call cotyledons. Cotyledons are the food stores which provide the food to get the first bits of growth going until photosynthesis can occur. Now there is a practical on germination that you need to know about, core practical. How would you investigate the factors needed for germination? And what you can do is you can set up this apparatus with 4 tubes, all with the same amount of cotton wool in, and all with 10 cress seeds in the bottom. Put cling film over each tube and control all the other variables to make it a valid experiment. And you leave it for 5 days. And you'll set it up as shown in this table here. One with water, room temperature and oxygen, one with no water but room temperature and oxygen, one with boiled water and no oxygen, and one with water in the fridge and oxygen. The results show that only tube 1 seeds will actually germinate. Tube 2 is no water, which means the seed coat can't get broken down and the enzymes getting activated. Tube 3 is no oxygen, as it was boiled and the oil stops the rest of the oxygen getting in, so there's no aerobic respiration going on, so the seeds can't respire and grow. And tube 4 is way too cold for the enzymes to work. So you need temperature, oxygen and water for seeds to germinate. You could also set up a tube like tube 1 but cover it in foil to block out all the light and see if that has any effect. Could you predict the results of that experiment? Well, actually, you'd still expect germination to occur because you don't need light to germinate. So you still expect the seeds to germinate. But I suppose if you left it long enough, you'd get the germination, but then you'd get death because there wouldn't be any light to continue the plant photosynthesizing once it had germinated. But it wouldn't affect the actual germination process.